Hi, my name is Tatsia Seamus Corlett. I just completed my undergraduate degree in biological chemistry at University of Toronto. I will first talk about why I'd like to study the origin of life at LC, about my past research experience, and finally present my project proposal. So why origin of life? Where do I begin? And I think for me, this is the book that started it all. It introduced me to the wonderful world of genetics, evolution, and the science of life. And I certainly didn't stop there. I kept reading any books I could find about life, evolution, complexity, the transition from matter to life. And then I moved on to reading papers for fun about LUCA, artificial life, hydrothermal vents. Origin of life is one question that I keep coming back to. Because I think asking questions about the origin of life is one of the most valuable, meaningful things we can do. And I can't imagine a more beautiful thing. Life is the greatest invention in the universe. And we don't know anything like it. It's a 4 billion year old planetary scale experiment that keeps generating new possibilities every day. And thinking about the origin of life to me is a constant reminder to appreciate the fact that we exist at all. And so why LC? LC is a unique place because there are not that many institutions dedicated to the origin of life. I know there's an initiative at Harvard, which LC is also a part of, and Santa Fe Institute does complex systems, which often overlaps with origin of life. And there's a Canadian university for origins-related research, but it's actually hard to find labs or institutes that are openly dedicated to studying abiogenesis, and that makes LC special. And I've had the opportunity to talk to Harrison Smith. I asked him about how it's like to work at LC, and then he's written papers with Cole Mathis at Santa Fe Institute. And Eric Smith is at LC. He's a wonderful guy. I love listening to his talk. And this Henderson Cleaves, right? He wrote one of the books that I listed earlier, A Brief History of Creation. So when I look at all these people who are affiliated with LC, I think it's almost like when I went to see The Avengers for the first time in the theaters. It really makes me feel like all these scientific heroes are coming together in this one place. So moving on, I'll talk about some of my research experience. And the first time I got involved in research, we wrote a review paper on the population analysis of space travelers. We looked at the demographic trend over the years in terms of which country they're from, their age, gender, etc., and also their bone density changes before and after participating in space missions. Then I volunteered at a lab, doing some wet lab molecular biology work. I learned some basic techniques like cell culture and running gels. And during the pandemic, I also worked on some theoretical work in organic synthesis, where we tried to decompose a reaction into several separate parts and then connect them together like Lego in order to find new reaction pathways. And because of the pandemic, I couldn't do any hands-on research for a while, so I tried to learn more computational methods to understand the origin of life and complexity. And I remember watching a keynote lecture by Mike Russell at a virtual conference called Artificial Life 2021, where he said, life is not mass action chemistry, it's a kind of autonomic computing. So I became interested in how we may understand life as a computational phenomenon. Then I found a group that I currently work with at University of Toronto. He's Alan, my PI. Their Matter Lab is all about using AI models like GANs and neural nets for material discovery, searching the chemical space for useful molecules for organic lasers and battery electrolytes. And in my thesis project, I worked on diquats, which are redox-active organic molecules that you can dissolve into water for this new type of battery called redox flow batteries. And the group has developed a generative model called Janus to predict structures with desired properties. And my job was to actually synthesize some of these molecules and to test their electrochemical properties. So fortunately or unfortunately, since the COVID situation got better, I mostly worked on hands-on organic synthesis and not so much on using AI. But working with this group taught me a lot about the methods in chem informatics and how people use AI to search through the chemical space for new molecules. And speaking of the chemical space, I want to dive into my project proposal titled No Code, No Life, The Origin of Code and the Expansion of Chemical Space, which was partly inspired by this tweet from Sarah Walker, astrobiologist at Arizona State University. She asks, what's the shape of life in chemical space? So chemical evolution is a popular idea that describes the process of simple reaction networks gradually expanding its repertoire and becoming complex. And people believe that if these autocatalytic sets can sustain itself by catalyzing the formation of its own components, they may have been able to reproduce, fight with each other for resources, and go through natural selection. And people have tried to simulate the evolution of such ancient metabolic networks from simple geochemical precursors, where evolution of coenzymes could lead to stepwise expansions of the size and complexity of these networks. So you can imagine these simple primordial metabolic networks occupying a tiny region within the chemical space. Then, through chemical evolution, the system gradually gains access to a greater variety of molecules. And let's say that there are specific molecules that reaction networks need to access for the origin of life. My concern is that no matter how long you wait or how lucky you get, reaction networks may only become marginally complex, because the types of molecules it can make will always be constrained by energy barriers and short-range interactions. And the key to explore the space more effectively, I think, is the code. And when we look at modern life, we see the significance of having a code. You have digital 1D molecules coding for analog 3D molecules. In reaction networks without a code, complex molecules like these would have to be constructed step by step, adding the components one by one. But when you have a code, complex molecules can be printed from lower dimensional templates. And I see two significant advantages. Right? First, you gain the ability to search for new polymer sequences via mutations. So the burden of evolution shifts from 3D functional molecules to the 1D information-carrying molecules that code for them. And second, it allows long-range interactions between molecules, not only in space but in time, because progress in chemical evolution can now be saved. A written language is born, which is indispensable for robustness and evolvability. And I don't know about you, but to me, that sounds familiar. So you can imagine this system now has access to a wide range of polypeptides. And what's even more remarkable about this is that these polypeptides can then act as enzymes that catalyze new reactions, like channels through chemical space. Enzymes allow the system to access molecules that are otherwise inaccessible. And you see this beautiful story of chemical evolution. Proteins are life's solution to make a wide range of catalysts, which then enables life to access a vastly larger space of possibility. So maybe in a distant galaxy, there are other instances of life composed of different molecules, but I think they must have this separation of digital and analog molecules and a code connecting the two. And I don't think life is possible without a code. No code, no life. And that's why I want to study primordial protein nucleic acid interactions, to gain insights into the origin of code. How did molecules start to talk to each other? That's the fundamental question I'd like to address. And one potential subject is ARS, or aminoacyl tRNA synthetase. In modern life, there are 20 ARS families, one for each amino acid. ARS first activates the amino acid by adenylation and attaches it to the corresponding tRNA. So in effect, ARS has defined the genetic code by physically linking the two molecules. So studying the evolutionary history of ARS enzymes may shed light on what the early protein nucleic acid interaction may have looked like. And in the history of protein evolution, it is widely believed that basic and aromatic amino acids came much later. And people have tried to simplify modern proteins using a smaller set of early amino acids, or even non-canonical amino acids, in a process called deconstruction. And there's some evidence to suggest that primordial proteins relied on metal ions like potassium and magnesium to bind nucleotides, and that high salt concentration can help primordial proteins fold properly. So we can ask, what's the simplest minimal ARS ancestor that can fold and bind nucleic acids, possibly with the help of such cofactors? And it is generally thought that ancient proteins were multifunctional or generalist proteins that bound a wider range of substrates with lower catalytic efficiency. And it reminds me of slime molds, slowly figuring out the shortest paths to nearby nutrients. First, you search a wider space of possibility, but as the system evolves, it becomes more specialized. So does the ancestral ARS bind a wider range of substrates? Can we rewind the clock of evolution, reconstruct the ancestral ARS to see what kind of reactions it can catalyze? In conclusion, the evolutionary lineage of ARS is connected to several key questions about the origin of life, including the origin of code, amino acid metabolism evolution, and primordial protein nucleic acid interactions. The reconstruction and characterization of primordial ARS enzymes would be a fascinating research topic that could be investigated from multiple perspectives. Thank you.